Alrighty. Hello, hello. What is up, Nathan? Hey, Hashi. Hope you're doing good tonight. If you got questions, just feel free to throw them at me. What's up, Rebecca? Let's see, what's your question? Why does porosity increase by adding clay particles? If clay is smaller particles, won't that be less porous than adding sand particles? No, porosity does not increase when you add clay particles. I don't know where you got that from, but yeah, when you add clay particles, you definitely decrease porosity. I'm not sure if maybe you misheard that. So if you look at like these kinds of images. What's up, Nathan? What's up, Diaz? Got two Nathans here. So basically what happens, right, is your reasoning is correct, right? So you're saying, like, why does porosity increase when you add clay particles if clay is smaller? It doesn't increase. I'm not sure where you got that part from. That might have been just a maybe you read something. This is the diagram I used, right? So the pores in the soil will close up or decrease as you add in clay, which are smaller particles, which can kind of fill in the pores between the larger particles, like sand or silt. So I guess maybe all you need to know is that it doesn't. It actually, porosity decreases. Here's a little graph. All right, so as clay amount increases, you can kind of see porosity decreases. Yeah, no worries. Thanks for asking. Thanks for stopping in. What's up, Aria? What is up, Onslaught? How are you? Anyone in the chat that plays Minecraft know any good Minecraft mod packs? I was looking up some mod packs for Minecraft. What's up, Amina? How are you? I was looking it up on CurseForge. I was looking at some reviews of this one, the RL Crafts, but apparently it's like really, really hard. Yeah, honestly, that one apparently was really hard though, so I wasn't sure if that would be good if it was too challenging. I'll take a look at it though. Yeah, the exam will be pretty similar to the one on AP Classroom. I would not expect it. The style of question should be pretty similar. Cover the same kind of material. Yeah, don't expect anything much of a departure. Pretty much right up that same alley. Ashi, the question on the study guide: which protein allows the cell to communicate with other cells? That's going to be a glycoprotein. So if you look at the cell membrane, let's go back here. We go cell membrane. Let's pull up this one. This is an okay one. So with the cell membrane here, you've got, where is it? So here's a little protein that has a sugar chain coming off of it, or a carbohydrate chain. We call this a glycoprotein. We didn't really talk about these too much in the class, so I wouldn't expect you to like know a lot about these, but just kind of understand that one cell's glycoproteins, which is like a protein that's embedded in the membrane, and then what sticks off the top of it is a chain of sugar, kind of like a polysaccharide that sticks off the top. This is sort of almost like the way I described it in class was like, this is like an ID tag kind of for the cell that allows cells to communicate with each other. So they know like what each cell is and send signals. Yeah. So the term glycoprotein, I think, did I use it? I spelled that wrong. Yeah. So glycoprotein is any class of proteins that have a carbohydrate group attached to the polypeptide. You look at the function. Where's the function? Oh, there it is, functions. They have a lot of functions, obviously, if you look at this, but where is the... like this kind of thing, cell recognition, that kind of stuff. Yeah. What's the format of the test? The format is going to be, it's going to be broken up. It's all multiple choice, so there's no short answer for this one. It's going to be broken up into 4 parts. So what will happen is the test is going to be broken into 4 distinct parts, and each part will sort of we'll take a part. You'll be able to review any questions in that part. It is time limited though, but within the time frame for that part, you can go back to any questions you want to review them. But once that part's over and we move to the second part, then you can't go back to the other part. So essentially it'll just sort of be 4 distinct little parts that we'll take with like a slight break in between. So a little bit of different format than the last time, for sure. A slightly different format. And I think that the plan is that hopefully that allows you to be able to sort of pace yourself better by having it into parts, and you'll you'll be able to like revisit questions and check and review them before you submit. Let's see, Nathan, for the phospholipid tails inside the cell are hydrophobic, nonpolar, and the phospholipid heads outside are hydrophilic/polar. Yes, Nathan, that is correct. So if I go back to this cell membrane diagram, whoa, that's a weird one. That's not what we want. I guess we'll just stick with the one that we had here. This was not a bad image. So yeah, so notice like this is actually a good diagram because it sort of peels the lipid bilayer, right? It kind of peels it apart. So you get like a top layer and the bottom layer getting peeled apart. And so the long fatty acid tails that stick off of the lipid into the center of the membrane, the green center here, like kind of like the meat in the sandwich, so to speak, that is going to be the hydrophobic part, non-polar hydrophobic part. The glycerol heads, which are these pink dots, these pink parts of the lipid on the outer edges of the membrane, the bread of the sandwich, if you will, those are going to be hydrophilic. And they will be charged or polar. Ashi, how much time do you have per... Niharika, is there an answer key for the review guide? Yes, I can go over some of those too with us. I'll go over some of those answers in a second. The review guide, let me pull that up actually. I think I have it over here. So as far as the time, it's different per each part because there's different number of questions, but it's usually going to be a little less. It's going to be 50 seconds per question on average. So for example, one part will be 7 questions and you'll have 6 minutes to do that. And then another part is, I think, 10 questions and you'll have 8 minutes, etc. So they're going to be, they're about anywhere from 7 to 10 minute long each part. So pretty short. Overall, the test shouldn't be longer than about a half hour. I'm going to pull up the answer key to the cell bio review real quick so that I can put it up my screen to let people take a look at some of the answer keys. And again, if I miss your question in the chat, just retype it, because I know that it can go fast sometimes, especially when we have people, a lot of people watching. We don't have too many, we have 25, so hopefully it's not too bad. Let's see, Unit 2. What do we want? We want review key for cell bio part 1. Is this the right one here? Yeah, here we go. I do have the key, so I can, I obviously can't show the entire key all at the same time. Let me give myself a little more real estate here on my screen. I can't show the entirety of the key at the same time, but if you want to go over a particular answer, we can. Just let me know. I'm missing some questions right now, so I'm going to jump back. Megan, is the test the same as Mr. Gann's? Yes, indeed. All bio teachers have the same test. Amina, for this problem, suppose a cell is 18% glucose placed in, open up notepad. Do I have a notepad here? I like having a notepad open just so I can do some drawings for y'all. All right, so Amina, we're going to go over your question first. So you said for the question, suppose a cell is in an 18... Wait, Notepad? Does Notepad let you draw? Wait, Notepad doesn't let you draw? What's the window app that lets you draw? Windows app that lets you draw. Do I have to go to Paint? Paint, that's what I was thinking. Paint. Okay, so your question was, suppose a cell is 18% glucose and is placed in an 18% glucose placed in an 18% water. No, good question. No. So if it's 18% glucose, that means it's 82% water. And so for example, so if it's 82% glucose, it's 82% water. So H2O is 82%, then that means the glucose is 18%. So it has to add up to 100. And that is the cell that's placed into an 18% water solution. So water is 18. And that means whatever else, the solute, maybe it's glucose, maybe it's something else, is 82%. So these are actually reverse. These are actually opposites. So what would happen is the solution is hypertonic to the cell. So what would happen is we would say the solution is hypertonic to the cell, and we would expect water to move out of the cell into the solution. This is the cell. And this is the solution. So be careful with the percentages. You have to make sure it's talking about percent of salt versus percent of solute or percent of water. Onslaught, well, why are most of the deserts in the world found between 30 and 30? Found between 30 north and 30 south? Yeah, so here's the next thing. So when you have, here's our Earth, right? Here's our equator, 0 degrees. And then we've got our 30-degree band north, 30-degree north, and you've got your 30-degree south, right? 30 degrees south. That's terrible handwriting there. I forgot that there's a test in both bio and apes tomorrow, so we might have a lot of questions. 30 degrees south. Remember the Hadley cells when you get the sunlight coming in, right? Sunlight. What it does is it heats up the equator. And so you get heat. And what happens is you get hot air rising at the equator. And then that air cools and sinks as it moves away from the equator towards 30-degree latitude. And because it's cooling down, all of the precipitation falls back down to the equator. And so this is actually now, not only is it cool, it's also dry. This is dry air. So because it's, wow, it doesn't even look like dry once I do that. I gotta be a little bit better with my handwriting here so that people can understand what I'm actually doing. So this is dry. So that makes sense that the deserts would be at around the 30-degree north and south latitude, because this cool, dry air is dropping down at 30 degrees north and south. And all the rain is falling at the equator, which is why the equator is hot and humid. And the 30 degrees are dry. Right, so this is that's the answer to that question. So I'll just, in case, if you have questions about the review guide, I'm going to post this key. There's the key. So if you want to look at the key, feel free to take a look at the key. I just posted it right there for you. So take a look at that. And if anyone else in the chat, if you're asking about key, like the key, and other people see that people are asking about the key to the Unit 2 guide, can you relink them? Actually, I don't know if you can post links in the chat. Shoot. I guess I'll just have to relink it later. Yeah, I do have office hours on Friday, so, and I don't think they're completely full quite yet. Okay, so I'm coming back down to the bottom of the chat. I missed some questions, so if I don't answer your question, make sure to repost it. But I'm coming back down to the bottom of the chat now. So Mohammed, you're asking, if we get a question saying what is a subunit for lipids called, what do we say? Well, the subunit for lipids, you have two. So lipids have, this is a lipid, right? Here's this, and then, right, let's just take a phospholipid for an example. You would say the subunits are the glycerol head and the fatty acid tail. That's all you gotta say. Let's see. Ahana. Oh, thanks, Ahana, for answering Amina's question. Yeah, those of you who can answer each other's questions in the chat, it's super helpful for me. So, for example, Ahana, that's awesome. I appreciate that because it helps me, because I can't keep up sometimes with the amount of questions that are coming my way. And I don't want people to not get their question answers just because I missed it or I can't keep up. So yeah, people like Ahana, the real MVPs in the chat, appreciate you. And I'll also try and double check explanations just in case people make a mistake in the chat when they're explaining something. Are you talking about the answers to the practice test that I put, the student made practice test? I have some of those. So here is the student made That's a link to the student-made answer key to the practice test. What up, Adarsh and Mr. Chang? I saw you guys in the chat there. I saw you guys popping in. I appreciate you stopping in even though you don't have the classes here. A little office hours stream. So I linked the Unit 2 review guide, and I linked the student-made practice test answer key. I'm going to see. I think I missed some questions. Oh, Daphne, that's a question. So Daphne, could you go over why saturated lipids don't have double bonds and why unsaturated lipids have double bonds? So basically, the reason why, it's just a matter of fact. So when the word saturated just means there are no double bonds. They're only single bonds. And the word unsaturated just means there are no double bonds. So when you ask why saturated fats don't have double bonds, that's just the way we name them. That's just the way we name them. So I don't really know how else to describe that. So let me erase this real quick. Actually, is there just an easy clear button here? Just like clear. So I don't have to like sit here and erase things for hours. Because that's going to take forever to erase. Yeah, just do new. Don't save. So here's an example of a fat. Actually, you know what? It's probably easier for some of these for me just to go like saturated versus unsaturated. And just look up an image because I'll find an image that looks way better than what I can draw. Here, I always use this one. Right? So it's just the matter of fact. Saturated fats have all single bonds. Unsaturated fats have at least one or more double bonds, which you see there. So can I go over question 4 on the practice test? So question 4 on the practice test is this one. What is a chemical bond that involves sharing? Oh, okay. We didn't, so don't worry about that. Ahana, don't worry about that because we didn't cover this. We didn't cover bonding. So we might get into this more in the next part of the unit. So go ahead and ignore like 4 and 5 here. Are totally not on the test for now. What's up, Evelyn? Manu, can I go over passive transport? So passive transport basically has basically passive transport just means it does not require energy. So for example, like if I were to just sort of write something, so passive transport, let's make this a little bigger. Passive transport does not require energy, goes from high to low concentration, aka with the concentration gradient. These are just some facts. Is there anything else that you want me to, that you're interested in knowing? It can be, passive transport could include simple diffusion as an example. Some examples would be like simple diffusion is one example. Osmosis, facilitated diffusion. Those are examples. The only kind of the only kind of cell transport we learned that would not be passive would be things like endocytosis, exocytosis, and active transport. Let's see. Ria, can I go over how removing vegetation would affect a watershed? So essentially with the watershed thing, we didn't cover, I sent a message out on Schoology basically saying that you don't need to know watersheds for this test. We're going to learn that more next unit. But essentially, the key thing to remember is that just for future reference, when you remove vegetation from a watershed, what that means is you probably will increase the erosion. And so what happens is because now you have more erosion, more topsoil is transported into the waterways, which can create more sediment and make the water more turbid, aka more cloudy, which can have an impact on the amount of light in the water, which can harm photosynthetic organisms and ultimately cause issues with the rest of the food web. So typically, removal of vegetation in the form of agriculture, when you create these big, large fields and you remove native vegetation to make cropland, oftentimes erosion increases, which has these detrimental effects of harming the watershed through runoff of topsoil. And it can also bring things like fertilizer too, which isn't so good. But that's kind of the general rule. How does endocytosis and exocytosis use energy? Powerful people. Because when you do endocytosis and exocytosis, you have to change the entire structure of your membrane. So if you look at an image here, right? What's happening in this picture is notice the entire membrane is changing shape and like pinching off a piece of the membrane and then moving inside the cell. So just the process of pinching off a piece of the membrane requires a lot of energy to transform the surface of the membrane. So it's just the sheer nature of how the process works requires energy. And you don't need to know much more than that. So it's not really necessary to go into too much more detail. Wait, so someone... So Mohammed, let me get your question. Are we supposed to memorize the different types of proteins, lipids, etc., and what they do, or should I just know what the biomolecule does in general? You should definitely know what the biomolecule does in general, but you should know a couple examples, right? So for example, you should know something like for proteins, you should know a couple examples like hemoglobin, maybe collagen. What else? Hemoglobin, collagen, like antibodies. You should definitely have a few examples that you kind of know off the top of the head, just to help you, because I can imagine there being a couple examples on there that you should know. But like hemoglobin, collagen, and antibodies are good. And then as far as like lipids, you should just kind of know like phospholipids, which are what make up the cell membrane, and you should know triglycerides, which are the fat that that you find in food or animal products at least. And then for nucleic acids, you should just know DNA and RNA. Those are really the only two. And then for carbohydrates, you should know cellulose, starch, glycogen, and glucose. Cellulose, starch, glycogen, and glucose. And I believe those are on, if I remember correctly, yeah, right here, boom. Right, so on this key of the Unit 2 review guide, here are the carbohydrates you should know, right? And a little bit about their information there. Some examples of lipids are here. So yeah, use the ones you see in the study guide. I think that'll be helpful. Mr. Chang, nice. That is good news. Get your associates for transfer around spring 21. Nice. That's awesome, man. I love to hear that. Megan, you should know hemoglobin and collagen. Yeah, you should just kind of know. Hannah, I'm going to answer your question in a second about the rain shadow inland continent effect. So I'm going to do Hannah's question about the rain shadow effect and inland continent. And then I'm going to answer Amina's question about vesicles versus vacuoles. Although I think I'm looking at Nathan Diaz with some nice answers in the chat there. That's really good. All right. So Hannah, rain shadow effect and inland continent. So do you remember... Let's talk about inland continent first. So, do you remember when I was talking about Ireland versus Wisconsin? So if we look at like Google Earth and we wait for it to load, when you have a place, so just in general, when you have a place that's on when it's in the middle of a continent, okay? So I'm going to zoom in. Here's the United States. Wisconsin is somewhere over here. Here's Wisconsin, okay? Wisconsin, you would say pretty much in the middle of the North American continent, right? If you trace this over, here's Ireland. Definitely not in the middle of a continent. It's an island. So if I zoom out just a touch more, does this allow me to put the latitude, longitude bands on here? No, it doesn't. Anyway, so basically, if I zoom out, Wisconsin is here. Ireland is here. Ireland is actually further north than Wisconsin. It's actually closer to the North Pole than Wisconsin. So you would imagine, well, because it's higher latitude, closer to the North Pole, it should probably be colder. But in fact, it is warmer on average. Its climate is more warm because it has the moderating effect of the ocean around it. And particularly the Gulf Stream, which is coming across the Atlantic this way, bringing warm water from the Gulf of Mexico. And so Ireland is actually relatively warm for its location. Whereas Wisconsin can get brutally cold because it does not have an ocean around it to keep its temperature moderate. So it has really, really cold winters, even though it's further south. Right? So that's the idea of being inland on a continent versus on the coast. Or being surrounded by a large body of water. The rain shadow effect has to do with mountain ranges. So I'm going to pull up, let's go back here. So a good diagram, we'll just sort of look at one. This is a classic one. So the rain shadow effect doesn't really have to do with temperature. It has to do with changes in precipitation. So remember, climate is the average rainfall, average precipitation, and average temperature of a region is its climate. So when we talk about climate and we talk about rain shadow effect, we're talking about the effect of average precipitation. So when you have a mountain, on one side of the mountain, we call it a rain shadow because the mountain blocks rain clouds. So when the clouds moving inland reach the mountain, all of the rain is falling on this side. And so what happens is a very dry region on the other side. So even though the left and the right side of this mountain range, let's say this is west and this is east, are at the same latitude, and you would expect maybe they would have the same precipitation, they indeed don't, because this mountain range sort of causes all the rain to fall here and none here. So this is a much drier climate than this side. Make sense? Let me know if that wasn't clear. I kind of went fast. Adarsh, if you can hold your question for a little bit later. I think, people are kind of wanting to get their questions answered for the test tomorrow. But I always love your questions. You have always got good questions. Amina, do humans produce 10 or 11 amino acids? Don't worry about that. That is not an important question for this test. If you really want to know, you can just Google it. I'm seeing a lot of 10s, 11s in the chat. I don't know. I actually don't know the answer off the top of my head, but if you just Google it, I don't know. It's not that important, obviously, if I don't even know it off the top of my head. You got it, Hannah? Okay, good. Because those are kind of important things in regards to average climate. And I know I talked fast, so I'm just trying to keep up with the questions. It seems like a real issue here. I mean, what did a Google search not like resolve the issue in the chat? How many essential oils? How many essential amino acids? One, two, three, four, five, six, seven, eight, nine. Maybe the answer is vague because there's these conditionally non-essential amino acids, right? Depending on your diet. Right? If you have a healthy diet, I think there's 9. But it doesn't really matter. Is the function of glucose just like being the energy source for humans and plants? Glucose has 2 functions. So glucose, oops, I don't want to save. So glucose has 2 functions. So let's say I have a glucose molecule. Boom, 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 boom. Here's glucose. It's got 2 functions. One, it can be used in cellular respiration, like you said, just to produce a form of energy called ATP. And that's useful for plant cells to function. So that's one function. The other function is it can go and it can turn into something like cellulose. This is another function. It can turn into cellulose, which is what makes up the cell wall. So cell wall structure, rather. So it can be used immediately for energy in this path. This would be your immediate energy path right here. But on the other hand, it can be used to create polysaccharides like cellulose, which can be used as part of like a structural component to a cell wall. It can also be used to make glycoproteins. So its only function is not to make ATP and energy for the cell. It does have other functions, but by and large, its sort of, I guess, quote unquote, most important function or most vital function is this production of ATP, because ATP is the energy currency of the cells. Ahana, could you please tell me what the polymer and monomer / subunit each biomolecule have? I think that shouldn't that just be in the study guide? Okay, monomer. So protein is the polymer. How can I write this that makes sense? Protein is the polymer, and amino acid is its monomer. Monosaccharide. These are just general terms, right? Monosaccharide. Excuse me. Polysaccharide is the polymer to monosaccharides. This is for carbohydrates. And then you've got nucleic acids is the polymer to nucleotides. And then you've got lipids are composed of the subunits glycerol and a fatty acid tail. That's it. Bum, bum, bum. What else? Onslaught, I got your question, reposting question again. For the APES study guide, I didn't actually draw the hotspots, arcs, and convergent divergent plate boundary on the map directly. I just labeled them. Is that okay? Yes, that's okay. But I will tell you it's possible for you to get a question on the test tomorrow that shows you a map with some plate boundaries, and you should kind of know what kind of boundary you would expect to see there. So let's see. So let's see, what's a good map? What's a good map? What's a good map? Something simple. That's really small. Let's go like this one or something like that. So I might give you a map like this, maybe like this, but you should kind of know where some of the key boundaries are. So for example, like you should know that in the Atlantic, this is the main. So remember when I was lecturing on plate tectonics, I kind of highlighted some key examples. So for example, in the middle of the Atlantic, you should be aware that there is a divergent boundary. And this is where two plates are spreading apart, creating a ridge at the bottom of the Atlantic Ocean. You should also kind of know that along the west coast of North America, you have a transform boundary where you have two plates sliding past each other. This is the San Andreas Fault, right? And that creates earthquakes. And then you should know on the western side of the Pacific, it's kind of got cut off on this map. But let me find a better map here. Maybe this one's a little better. On the western side of the Pacific, you have the Pacific Plate, which is a convergent boundary where you have one plate sliding under another plate, which generates earthquakes as well. And you can have some volcanic activity. So the ring of fire, quote unquote, is the ring around the Pacific Plate where you have subduction occurring, and you've got a variety of volcanic activity happening around there. So those would be some what we would call hot spots. I have to grab some water. I'll be right back. And looking at the chat, it looks like most people are getting their question answers by each other, which is really awesome. That's kind of what the chat's supposed to be like, which is great. Good stuff, good stuff. Yeah, so shout out to Ahana, Nathan, Manu, Amina, Advait. Good stuff, guys. If I missed anyone, sorry. Onslaught, just to kind of go back to your question about what are the other geographic features that go with transform faults? There really is none. It's just earthquakes. That's it. There's no ridges, no trenches. Yeah. So divergent boundaries can form earthquakes, volcanoes, and I think ridges. No. So divergent boundaries, you will not get earthquakes at divergent boundaries. If two tectonic plates are spreading apart from each other, you're never going to get an earthquake. In order to get an earthquake, you have to have two plates that are moving past each other in some way or another, because when they're moving past, they can build up friction and then slip, and that's what causes an earthquake. So for example, you could have an earthquake at either a transform or a convergent boundary. So either the plates are sliding past each other laterally, which can cause an earthquake, or one plate is sliding under another, which again can create friction as it's sliding under. But at a divergent boundary, you're only going to get volcanic activity. So of all the boundaries, I would say, just sort of the way I like to think about it, is the worst boundary to live at is a convergent boundary, in my opinion, because you can get earthquakes and volcanoes at those boundaries. Whereas the other two, it's either or. So, and the one that the boundary that tends to be the least sort of interesting from a topographic point of view is transform. They don't really have any major geographical feature like a ridge or a trench that you would find at the other two boundaries. They just have a lot of seismic activity. What are some other questions in here? Saketh, cholesterol. Nice. Thanks, Saketh. Wait, so glycerol makes up the lipid triglyceride. Sasank, thanks for the answer. Nice. Thank you for answering Amina's question. Let's see. On the study guide where it says 3 geological features / processes per major type of, but you don't have to meet all 3 features. Oh, did it say that? Did it say, did it say 3? Well, I don't think there's more than, I mean, there's not really any other sort of unique feature. I mean, this says there's volcanic activity, but as far as I'm aware, there's really no volcanic activity at a transform boundary. So as far as you need to know for the test, is just know that transform boundaries, it's earthquake is the issue. Yeah, don't worry if you can't. Yeah, I don't really know what other features you could write there. Sure, yeah, you could say that, the erosion, but ultimately the key thing is that it's just seismic activity. All right, what else? What's up, Legend? No, this is not for freshmen. This stuff is all. This is AP Environmental Science. It's juniors and seniors. So just FYI, some people in the chat are not aware that I teach environmental science as well. So if you're a freshman taking bio and maybe you're not in my class and you aren't aware that I also teach AP Environmental Science, I also have a test tomorrow in my APES class. So what you're seeing when I describe some things, if you're like, that does not sound familiar, it's probably not for your class. So I don't want to make things super confusing. I don't want suddenly all of a sudden someone in Miss Lerner's class to go, I thought we were supposed to know about, convergent and divergent boundaries. What's up, Geeta? How you doing? Amina, what are microtubules and microfilaments? Those are examples of proteins that make up the cytoskeleton. So if you look at like a picture of the cytoskeleton, it will be composed of, what's a better picture? It'll be composed of 2 kinds of proteins called microtubules and microfilaments. I don't know if I can find a really good picture of that. Is this a good picture? Yeah. Right. So microfilaments and microtubules make up the cytoskeleton. What else? Any other questions in here that I'm missing? We were talking about cholesterol earlier, and it looks like, yes, it stabilizes the membrane. It makes the membrane more rigid. So the more cholesterol that you have in your cell membrane, the more rigid the membrane becomes and the less fluid it is. So actually one of the reasons why if you have too much cholesterol, it can make your arteries rigid, more hard and less elastic. And that's not good for your long-term cardiovascular health. It can actually lead to the arteries tearing a little bit and causing things like heart attacks and strokes later in life. JLAX, are cytoskeletons only found in cells without a cell membrane? No. A cell without a cell membrane is not a cell. A cell without a cell membrane would be like a house without walls, right? It just wouldn't exist, right? You need the sort of boundaries of the thing to establish that it is a thing, right? So without the cell membrane, you don't have a cell. Oh, Legend, the blue and orange flashcards. Those were, what were those for? The problem is it's online learning right now, so no, they're not getting any colored paper from us. They're not getting any paper, so they're not going to get them regardless. Wait, can someone dumb down the motor protein's purpose, because I searched it up and I don't understand? Yeah, so Ashi, I can help you with the motor protein. A motor protein, all that a motor protein is a protein that carries a vesicle. So, okay. So let's find a good image here that we can kind of quickly hit on. Okay. So here's a very basic diagram that I think can serve us. Oops, didn't load. Okay, you can kind of see that, right? So let's say that you have a vesicle. Okay, here's our motor protein, right? This right here, let's say this is a vesicle. And remember I talked about vesicles in the context that they are little bubbles of membrane that carry things, aka cargo. So let's say that there's some proteins in here. These red X's are proteins, right? And the cell has made proteins and it wants to secrete these proteins out of the cell. So the proteins were made through the pathway to make proteins, right? The DNA in the nucleus is read for the instructions. The ribosome takes the instructions, makes the protein. The protein is then sent to the rough ER, where it is sort of modified and put some finishing touches on it. The ribosome then sends it to the Golgi apparatus. And then the Golgi apparatus is going to package the protein into a vesicle and want to send that vesicle out of the cell. Now, how does the vesicle get to the cell membrane from the Golgi? It is carried by a motor protein like this along the cytoskeleton, which is, it says cyto, you can't read that, it says cytoskeleton. I think that says filament or something. And it literally walks. It walks back and forth, and it carries this big bubble of membrane towards the cell membrane. And eventually when it reaches there, it merges with it and releases it. Do you need to know this for the test? No, you don't actually. But this is just one example of a kind of protein, right? So we talk about proteins as a biomolecule and they have all these different functions. This is a example of a protein. Just an example. They're actually really kind of cool. They're kind of cute little things. I'd recommend if you watch an animation or something of them. They're like little workers inside of the cell carrying things back and forth. Let's see, anything else? Yeah. What do carbohydrates on the cell membrane do? They're a way for cells to communicate with each other. Think about the carbohydrates that stick off of the cell membrane as like ID tags, or ways in which cells can communicate with each other and send messages and signals. Amina, can you explain the process/order in which the organelles work? I think are you asking to explain the process/order in which organelles work to make proteins? Because that's a broad question. The process and order in which organelles work. There's really no order. Like if you're talking about the entirety of the cell and all of the organelles, there's no order in which they work. They're all kind of doing their jobs simultaneously to help the cell survive and carry out its function. When we talked about the order of organelles working, we were talking about the order involved in making and releasing a protein from the cell, which is essentially what I just talked about when I was explaining the function of these motor proteins. Sanjay, can I quickly say all the vague topics that we covered that'll be on the test tomorrow? No. Actually, I can't say all of them because there's too much. Just go to where is it good? I'm going to start closing some of these tabs. Things are getting a little out of hand here. Go to if I can open up. Just there's a link in there. If you go to Unit 2 Cell Bio and you open up this document, it's basically all of 1A, 1B, 2A, 2B, and 2C. So everything you see here from 1A to 2C, this is on the test. That's the test. Manu, I don't know if it makes sense to do the Kahoot because the Kahoot was, we already did it in class. So I don't want to play the Kahoot again because we went over it. On Slack, could you copy and paste a picture for the graph of the Earth's atmosphere? When you say a graph of the Earth's atmosphere, you're talking about the change in temperature? The temperature profile, like this one? Are you talking about this kind of a graph here? Yeah, I mean, the key is that you just know that there's like this weird fluctuation that occurs in the first couple layers before finally like really reducing in temperature. Or I mean, increasing in temperature rather. That's fine. What's up, Hackswale? How are you? Amina, does the practice... Gita, there's your question. Gita, are we allowed to do the study guide online and use pictures from... Are we allowed to do the study guide and use pictures from the internet? So I would say... I'll say this about the study guide. I think ultimately, I'm going to allow it to be your call. But ideally, for some things... just make sure that you understand what the diagram is. Just copying and pasting it can sometimes... I would say yes, it's fine. Just make sure that that you at least still understand the diagram. because sometimes hand drawing things helps you remember it. But I'll leave it up to your discretion because, you know, it's an AP class, so it's your call. I'm not going to make you do something in particular just because I think it would be better for you, because you can make that call on your own. So. There was another question I wanted to ask answer really quickly. Oh yeah, Muhammad wanted to know the differences and similarities between prokaryotes and eukaryotes. That's kind of a big question, because I think there's a lot of things that you could write for that. And what I think if you just look up a chart for this, find like I did. Yeah, like this is a good enough one. Just look over this. I think this is pretty good. Just look over this so I don't have to like spend too much time. Axel, I don't know what is that? Xi Jinping? I don't know who that is. Oh, the General Secretary of China? Oh, the Communist China. I don't know. I probably not. I bet he would probably beat me up. I don't know what his combat background is. He's the president now. Is that what the name is? They call that because it says General Secretary of the CCP. General Secretary is also president. Oh, but he's also the president of the People's Republic of China. Okay. Let's see. Ahana. Nice. Thanks, Ahana, for answering questions. Thank you again for those of you that are still sticking around and answering people's questions. By the way, I always recommend, like, as a way to study, is always good to try and explain things to people, because it forces you to sort of think about how to describe a topic in a way that someone else will understand, which is always like which can be like a really difficult thing to do. And if you can do it well, it's a pretty good indicator that you like have a really good understanding of the subject, or of the topic at least. So like the way I think about it is like there's like kind of like 3 levels to understanding something. There's like level 1, which is basically being able to like recite something, like answer a simple question about it. Level 2 is being able to like answer like a complex question that requires like multiple parts together. And then level 3 is being able to explain something to somebody in a way that they can understand. And you always want to shoot for that level 3. Ooh, rip Onslaught. I hope you didn't lose a lot of progress. So Amina, as far as the questions on the test, they're going to be worded a little bit differently. I would say that the questions on like the practice test, which I just lost. Remember, the practice test that I posted was more of a student-made one. And because it's student made, it's not going to be worded the same way that questions that are written by, myself or other teachers are worded, because we typically write questions in a little different way. And sometimes the questions that we rewrite are a little bit more, are usually more challenging, right? And so what you could expect is a little more challenging style of questions than the one you saw on the practice test. Think about the kinds of questions that you had on the ecology exam, that style and that level of difficulty, kind of expect that level. So again, it's going to be more challenging than the practice test. Yeah. Okay. Oh, the review. Well, the review, yeah, the review is not really the kinds of questions on here weren't really like multiple choice style questions, right? These were more open-ended questions. And so what you would expect is like the questions are going to be, oh, these got all messed up at the bottom, these labels. That's okay. I wouldn't ask you to label a cell on the test. You should know what some of the organelle's functions are, but I wouldn't ask you to label it. So that's actually okay that that's messed up. But yeah, there's no questions in here. So it's just content, it's just material, fill in the blanks, charts, etc. So the questions are going to be worded more like multiple choice. So it's hard to really give you an exact answer there. I would say maybe the ones in the formative assessment might be a little more similar to the test. Let's see. I want to see. I think there's any other major topics we should cover. Haxel, do you think you could beat him up? Based off what I saw from that one clip you sent me, I think you got a real shot if you can get that in. If you can get that one, you've got that one punch. One of the Quizlets, there was a question about someone who was lactose intolerant. Would we have a question like that? So if you, yeah, the lactose intolerant one, like I don't think you actually had to know exactly about lactose intolerance to answer the question. Let me see. If a person who is lactose intolerant consumes milk product, explain using your knowledge of enzymes what happens in their small intestine. So based off of the question, you could kind of infer, right? So if they're intolerant of lactose, right, which is found in milk, what do you think will happen in their small intestines? You could say something like the lactose probably would not be digested properly because they don't have the right enzymes to digest it. So you could have gotten a little bit about it. But
a person who is lactose intolerant cannot process the lactose.
Oops. Okay. A person who is lactose intolerant, right, is basically they don't make the right enzymes to break down lactose. So there's an enzyme called lactase that is meant to break down lactose, and that's not occurring in those individuals who are lactose intolerant. So when they drink milk or eat cheese or any sort of product that has a milk product, that lactose causes them some digestive issues, and they tend to have like upset stomach, like diarrhea, gas, these kinds of things. So they tend to stay away from it. But if you're not lactose intolerant, you have an enzyme called lactase which breaks lactose down, and then you're fine. Yep. Yeah, thanks Amina for answering Ahana's question. Semi-permeable means some things can pass through, but others can't. Yeah. And Manu, good answer. Nathan, is the formative assessment practices because it isn't accessible to others. It shows a grade. Wait, formative assessment 2A/2C. Let me look at that right now. Formative assessment 2A, 2C. Oops, it should say 0 points, ungraded. And let me see. You should be able to do it. The settings should be fine for you to do. You guys don't have access to this one? Students. Make that yes, maybe. Has a time limit? No. You can. Currently isn't taking submissions. This one, right? Cell Bio 2, form of assessment 2A and 2C. Oh, shoot. The submissions were disabled. Okay. It should be fine now. You should be able to use it now. My bad. Yeah. This thing with Schoology is there's so many little different settings. Like if you forget one little setting, the whole thing is ruined. Yeah, I messed that one up. Yes, you guys are talking about activation energy. You're right on. Yep, you're right on the point there. Shravan, did you want to go over enzyme graphs? Sure, let's do that. Let's do it. Enzyme graphs. Let's take a look. So maybe this is a notepad situation. So you basically have, there's basically, let's just say 3 different kinds of graphs, right? So you have one graph. Let's say this will have temperature. And you have, let's say, 0 degrees up to like 50 degrees Celsius. And then on this axis, you have, let's say, activity of enzyme, maybe. Enzyme activity. Abbreviate enzyme, ENZ. Enzyme activity. So an enzyme typically has an optimal temperature, right? And so let's say that this is the graph that you see. It's like right, this is the graph. What this shows is that the peak right here is when the enzyme works most effectively. And if I trace this down to the axis, let's say this is like 40. So this time, the enzyme clearly works best at 40. As it gets too cold, the enzyme's effectiveness decreases, right? And as it gets too hot, it also decreases. We call this denaturing. And that means the enzyme no longer can do its reaction. And so it changes shape and its function stops. Then you have pretty much the same thing. So if I just like copy this and paste it and move it over, and except delete temperature, I can almost do the same thing and just replace it with pH. So it basically it's the same thing as temperature, but instead we have pH. Wow, that ended up being actually a lot longer than just redrawing the whole thing. Right, so let's say we have pH of 1 to 10, and this is pH. Goes up and down. And so let's say here's the optimal. This would be, 5. Oops, not degrees, 5 pH. So in essence, the enzyme's activity, again, has an optimal pH of 5. And then the last graph basically looks like this. Again, you have enzyme activity, the same on this side. And instead, here we have number of substrates. Number of substrates. So let's say this is like 20 and this is 0. So it's pretty obvious, right, that as the, or let's just say this is 200, just to make it more realistic. So as you add substrates, which is the thing, the enzyme, the molecule or substance the enzyme is using for its reaction, as you increase the number of substrates, there comes a point right here we call the saturation point. And that point is when the enzyme can no longer work any faster. It's going as fast as it possibly can, right? And so let's say that number, if we trace it down, maybe that number is, 70 or whatever. Right, so those are the three things. Enzymes, and again, this same thing over here, I forgot to write this. On either side of the optimal, you get this denature. Amina, can I not have the 1, 2, 3, and 4? And no, those are really hard. I hear you. And those are the questions that we add to make it, to bump up the difficulty level of the test a little bit, to make sure that students can put more than one thing together at a time. Because when you ask a bunch of questions that are what I would consider like lower level questions, like just easy recall questions, like, which of the following is the enzyme that is, which of the following is the biomolecule that is involved in speeding up chemical reactions, right? Proteins, carbohydrates, lipids, or nucleic acids. Those kinds of questions are very simple and straightforward and just sort of like recall / memorization kinds of questions. When you throw in a question like a 1, 2, 3, 4, now it requires students to be able to connect multiple things together, even though it is more confusing. So those are kinds of questions that I put in there just to sort of separate out and see how really well people know some of this stuff. But I do know that they are hard, and I understand your... Oh, you want 1, 2, 3, 4 instead of 1, 2, 3, 4. Interesting. I've never heard someone say that. Does anyone else agree with Amina that if I used number 1, 2, 3, 4 versus Roman numerals, it would be better? This is the first time I've heard this. Is it confusing because is it hard to see, like when you see the Roman numerals, do you not know how to read Roman numerals? Or is it just that Roman numerals kind of look the same? Oh, you keep skipping over the lines. You know what? That makes sense. Use lowercase Roman numerals because they have dots. You know what? That's funny. I've never heard anyone say that, but now that you bring it up, it kind of makes sense, right? Because like a 1, I mean, it is a lot easier if you think about it. Because a 1, 2, 3, 4 versus 1, 2, 3. They definitely look a lot more different. Or just use like shapes. Do like circle, square, triangle, star. Although that circle kind of looked like a... No, I don't. There's no way for me to do shapes because I can't really put that into like a test. But we could do this. It's too late now, though. I can't go back and change it for this one. But for the next test, maybe we'll do a poll in class. I'll do a poll for this one. Lowercase, so you would do, Adarsh, you'd say like I, and IV. The problem, Nathan, with A, B, C, D is the answer choices are already A, B, C, D. So it has to be different than A, B, C, D. Right, like ultimately the actual answer you pick is either A, B, C, or D. So if it's like A only, B and C, I guess you could. I just don't want to get too confused with all the letters. I don't know. That's interesting. I never thought about that just from a format perspective. So, okay. I'll bring it up. I will bring it up. I'll bring it up with the rest of the teachers and see what their thoughts are on the situation. Does anyone have any other questions? Adarsh, yes, it sort of died down a little bit now, so you can ask away. You can ask away. Oh, Nathan, don't feel too bad for Adarsh. He's not even in APES. He's a former bio student of mine from a couple years back. Adarsh, what are you? You're taking physics, right? AP Physics. Mohammed, how many questions on each part? I think I want to say it's 7, 10, 8. 7, 7, 10, 8. So there's like 32 questions. Manu, the function of steroids. No short answer, Nathan. Just multiple choice this time. So the function of steroids are a type of hormone. So what steroids do is they send signals from one part of the body to another to cause some sort of change in the body or some sort of response. So steroids go from one part of the body to another as a signaling molecule, as a message. Adarsh, I'm going to answer your question in 1 second. No, I didn't forget it. Will we have to know the product that an enzyme and substrate make? No, I would tell you. Amina would tell you. Format of the test, like I said, it's broken into 4 parts, four short multiple choice sections. With each section has anywhere from 7 to 10 questions, and there's 4 parts for a total of 32 questions. Manu, you do not need to know the different types of RNA. You don't. Gita, can you talk about 4.3 for APE study guide, which talks about the primary soil nutrients, secondary soil nutrients, and fertilizer? Yeah, I can do that. So if you look up like, like primary soil nutrients, basically the way to think about it is that soil nutrients come in like three categories. You have like primary. We didn't really talk about this to that level, because if you look in the AP unit guides for from College Board, they don't actually have you go into the detail of knowing all of the micronutrients. But what you do need to know is the primary nutrients, especially, and those are the ones that you find in fertilizers, right? So fertilizer will always contain the 3 primary nutrients, which is nitrogen, phosphorus, and potassium, NPK. So the way I just remember it is NPK, and those are your 3 major nutrients or primary nutrients that are found in the soil, which is why fertilizer always contains those, because those are what plants need the most of. Secondary nutrients are going to be things like calcium, magnesium, and sulfur, which you see here, which are in a much smaller quantities. They will be in fertilizers as well, but the bulk of fertilizer is going to be these 3. And then you have like micronutrients, which are things, like copper, iron, etc., like these minor nutrients. So don't worry too much about all of those, but you definitely need to know the 3 major nutrients: nitrogen, phosphorus, and potassium. They are the limiting factors for soil, or for plant growth in soil. And then fertilizer is usually going to provide those 3 that you can mix in with your soil. Manu, you do not need to know the properties of water. Not for you'll need to know them for the second part of the unit. That's actually kind of what we'll start off with. The only endings you need to know, Amina, are OS and ACE. OS for sugars and ACE for enzymes. That's it. Daphne, on the 6th problem in the Cell Bio 2 formative assessment, it says that in a hypertonic solution, water will enter the cell and the cell will swell up. I thought it was the opposite. It would be. Let me look at the question. Wait, which formative assessment? 2A and 2C. The 6th problem? I'm trying to find... I don't know which question you're referring to, Daphne. I'm not sure which question you're referring to, but the answer is yes. If the solution is hypertonic to the cell, then water will go out of the cell. So it could have been maybe an incorrectly worded question. It's totally possible that the question was worded wrong or it was incorrect, because if a solution is hypertonic, Wait, is this it? Hypertonic solution. A solution whose solute concentration is less than the solute concentration inside the cell. That's false. This should actually be, this is not written correctly. These should be swapped. A solution whose solute concentration is greater than the solute concentration of water will enter. Oh yeah, this is wrong. Hold on. I gotta go to edit this real quick. Yes, you did find an error. You did find, oh shoot. You did find an error here. Daphne, you found an error. You got an error. Let me fix it because that is an issue. Wait, I can't edit it in here? How do I edit this? No, I need to edit the actual answer, the actual text in the answer. Oh, here we go. I have to edit it up here. Okay. A solution's no. A solution whose solute concentration is greater than the solute concentration inside the cell, water will leave the cell and the cell will shrink. Okay, and then a solution whose concentration is less than the water will enter the cell and the cell will swell. Right? Because if the solution whose solute concentration is less than inside, the water should go into the cell. Yeah, so then we need to change the ordering down here. A solution whose solute concentration is less than the solute concentration inside the cell. Water will enter the cell. Oh, wait, what? Solution whose solute concentration. So this should be switched with this. And this should go here. Hypertonic. A solution whose solute concentration is greater than the solute concentration inside the cell. Water will leave the cell and the cell will shrink up. Okay, this should be fixed now. That should be fixed. So yeah, good catch, Daphne. Yeah, that was messed up. That was incorrectly written. Whew. So Adarsh, I gotta go back to your question. I like how you asked your question right as it died down, and then like immediately after you asked your question, it was just like a floodgate of other questions. Like everyone was just waiting for you to ask your question so they could wash it away in a flood of other questions. So how do we store the boundaries? You mean boundaries? How do we store the boundaries between introns and ectrons when splicing pre-mRNA to get mRNA? When you say store the boundaries, is that what you meant? Store boundaries? Restore the boundaries? The locations of the boundaries? So when you've got like, so here's your mRNA, right? And then you said like, let's say these red patches, these are our introns that we want to get rid of. Okay, so let's say we've got like 3 introns in our mRNA, right here, here, and here. Okay. you're asking the locations of the boundaries. Where is that info stored? I think it's I think it's like a machine, like a mechanistic kind of thing. Like I think what happens is there's like a protein. Again, I don't remember. We don't really teach this in bio, and the last time I learned about this would have been in college, so it was a while ago. So it's a little sketchy. But as far as I understand, the machinery, which is a variety of enzymes and proteins that come in, right, and snip, like, right, so you have like a protein, right, that kind of like some protein that comes in and it will sort of snip and snip these edges, right? I think there's also something that just immediately like connects this edge to this edge and just like stitches them together, and then it does the next one, cuts it, and then stitches that end to that end. So when you say like how does it store the info, I think it's all happening at the same time. It's like, imagine like you have like a piece of string and you sort of cut a piece out and then you just sort of stitch those two pieces together, and you sort of do this stitching process. It's not like you cut out the introns, now you have all these different pieces of mRNA and you have to remember, like which end goes to which end. I think it's all done at the same time. Oh, wait. Oh, your question. You mean how how does it know where the introns, where are the introns? Oh, like how does it locate them? I'm pretty sure there are sequences. There are like little sequences of RNA that act as like almost like little flags. It's almost like kind of like, you know how there's like the promoter region in DNA that sort of is the beginning where transcription will occur to create the mRNA in the first place that's on the DNA. There's like these promoter regions. I think there's something similar. Like there might be like a little area, let's say, you know, in the beginning of each intron and at the end, that's sort of just like some sort of like, hey, here's where the introns are, right? Cut from instructions are like cut from here to here, and the protein that's sort of reading this, right? This protein that's reading all this is sort of able to read these little snippets. That's my impression. Again, I'm not an expert right now at mRNA splicing because it's been a long time since I've remembered the actual details of it. That's true. Yeah, we did. We talked about splicing in bio, but we didn't go into the level of the mechanistic details of exactly how a cell knows which way to splice it. So I guess if we want to think about like how does a cell know which way to splice it, right? Obviously, it's going to be splicing it in a certain way because it's needing a certain protein to be made at that time. So in order to make that protein that's necessary for the cell at that time, it needs to make a certain kind of mRNA. So it has to splice the pre-mRNA appropriately. So there's going to be some sort of signal that would signal the cell to splice it a certain way. Now the question is like, where does that signal come from? And also so there might be, this is actually something I don't know the answer to now that I'm trying to think my way through it. So how does the cell know, or how do we know like which way to splice it at which time? Because it could be done multiple ways. What's the thing that signals the production of a protein? Maybe it's in that signals the production of a protein. So it's so you're getting into like gene expression a little bit. So when we talk about gene expression, you're talking about certain genes in the DNA being accessible to be expressed or transcribed by RNA polymerase to make an mRNA copy. Yeah, I don't know. I think this one I'm not as sure off the top of my head. Yeah. I mean, we can just try researching it together. How does a cell know how to splice pre-mRNA? We don't want the steps. This is one of those questions. Again, we might not actually know the answer. You might be like bordering on like knowledge that we're not quite sure how, like, you know, there are things, of course, that we don't know within the cell still. Manu, what is your question? What are induction, deduction, and arrow and shield models? Where is that question coming from? I don't know where that comes. Before living organisms can do. Okay, we know what splicing is. We know kind of how it works. We have known for some time that splicing is turned off by one of the large family of RNA helicases, which are energy-consuming proteins that move along the RNA like trains on tracks. They can displace items bound to the track, just like a cow catcher functions on a train. Okay. We know that an RNA helicase called PRP43 terminates splicing, but we have not known what RNA it moves along to terminate splicing. Identifying the RNA that this helicase interacts with is critical to understand the mechanism. Okay, so again, I think we're literally on the border of our understanding of how splicing works. Right, like this article. When was this article posted? Like a year ago. Okay, so this is like a little bit over a year old. RNA helicase not only helps in the production of messenger RNA for translation into proteins, but it also helps in the synthesis for ribosomes. That links the production. For full activity, though, PRP needs. Yeah, I don't know. Bye, Ahana. Have a good night. Could be something like that, a Darsh, that it's just sort of a proportion of expression. Right, so... yeah. I mean, this is some pretty complex stuff. This is like definitely stuff that I don't think I even learned when I was doing like some of my cell bio and molecular bio classes in college. But, you know, I did not go down I would say, like, to really have a good understanding of this, you would have to be like a graduate student in like molecular biology, and maybe even like post-grad stuff, like specializing in gene expression is my guess, to have a real grasp of this. So this is you're getting into territory here that's like well beyond my understanding. Like, I don't have the level of, I don't have the depth of understanding at this point. So you're wading me into deep waters here, and I'm not equipped. So I'm going to sort of leave it there, because I don't have, like, we could spend time looking into it, but unless there's a reason particularly to do that. Does cellulose make up the cell wall, or does it just give it more structure to make it stronger? Actually, you know what? There's one question I wanted to ask. I don't know if people are still in the chat or not. I know some of you are leaving. Some of you, I would like it if some of you who I know take good notes during class, would any of you mind sharing your notes with me? Like, if you have a Google Doc or whatever way you take notes, I'm curious to see how you all are taking notes during remote learning. Like, I understand how people take notes normally in class, obviously, because I can just see them. But like, I'm not really sure what's going on behind the scenes for you guys. But if anyone wouldn't mind, like, the more people that share, the better, so I can get a better look at different kinds of note taking. If you could just like share a Google Doc or email me something, I would love to look over. And the other good thing about that is if you have good notes, I can share it with students who are maybe struggling in the class, which would help them see what good note taking looks like as a model. So I know that there's some of you out there that take really good notes. So if you send them my way, I would be very much appreciated. Thanks, Manu. Thanks, Arya. Daphne, that's okay. I'll see if I can do my best. I'm sure your handwriting is not so bad that I can't read it. So are you still taking notes by hand then? Is that typically... So who takes notes by hand and who's taking notes on a computer? Because I know that those are... In class, I notice everyone takes notes, but that's because a lot of times you're not allowed to have a computer out. Although I do allow people to take notes on computer typically in my class. Google Docs, okay. Yeah, Daphne, I hear that a lot. People taking notes by hand, it tends to help them more. And I think there's studies on that actually. Like if you look at like research on handwritten notes versus typed notes in retention, I think it's shown. Let's see, researchers found that writing notes leads to significantly more short-term retention. When students were tested to see whether they remembered what they wrote, students who wrote their notes remembered more than typing. Huh. Is this a study here? I don't know if I believe just like an infographic from a .com website. Oh, then you look at something like this. Writing notes by hand might not be better than typing. This is the problem, right? It's like always like there's always multiple things. This is from... In 2014, news headlines heralded the findings of a study comparing the effect of taking handwritten versus typed notes on student performances. The study was titled "The Pen is Mightier Than the Keyboard." In this 2019 study, researchers replicated and expanded the earlier work by using the same videos but adding a group that took notes on an e-writer and a group that took no notes. The researchers also tested students on the content of the videos 2 days after watching the exam to examine the effect of different note-taking styles on retention. Interestingly, these researchers did not find a clear superiority of handwritten notes over typed notes. Maybe it's not so clear cut. So this is the conclusion of the study. Based on the present outcomes and other available evidence, concluding which method is superior for improving the function of note-taking seems premature. This was in 2014, though. Oh, this was a replication study. Oh, okay. So they did, in 2014, they found data that supported handwriting, but then they tried to replicate it 5 years later, and it was inconclusive. Okay. So it sounds like it might be inconclusive. Again, I don't know much about this study, though. Educational Psychology Review. Don't know this article or this journal. But hey, I think whatever helps you better. I think it's more of a sort of you gotta know yourself kind of situation. Yeah. Yeah, the typing thing, I'm trying to think. So I typically do typing notes because I can type just significantly faster than I can write. Like so much faster that I think the trade-off in retention is outweighed by the sheer speed at which I can like make notes. I can just include more things in my notes. So I tend to lean towards typing. You just highlight over the important stuff the night before the test. Yeah. Yeah, I don't know. I think when I think about notes, I find that the notes that I take ultimately aren't really that useful. It's how I use them later. It's how I like go back over them and then try and take my notes and start writing out like study guides or something, or turn it into some other product. But ultimately, I found that the only real way to remember something long term is to really spend a lot of time with it, basically. Like as a teacher, I remember all this stuff so much better than as a student, just because of the amount of like repetition that I've done. And also when you teach something, you really have to like know a variety of layers to answer questions and to explain it in multiple different ways. So I think ultimately, the way you take notes, typed versus written, is a very small percentage of your success. Like most of your success comes from what you do with those notes and how you study after the fact. It's kind of like like when you're thinking about like, oh, I want to be when you're like basketball, for example, like what kind of shoes you wear maybe has a little bit of an impact, right, on your how you play basketball. But ultimately, compared to other important things like the fundamentals of it, like how well you dribble, like your shooting form, like your endurance level, all those things probably are obviously a much bigger role. So it's like, in the end, probably not the most important aspect of remembering stuff. All right. Looks like we're kind of dying down here. What time is it? 8:55. All right. I'm going to play a game of Among Us before we end for the night. Oh, actually, Minecraft. Adarsh, I don't really have time tonight to jump into a full Minecraft thing. Maybe I'll do a stream, a Minecraft stream. Another time, but I was trying to get some feedback on mod packs. So I was looking at some mod packs. So I've always had a problem with Minecraft is like when it's too open-ended and there's not like a little bit of something to do, like a directionality to go, I kind of struggle. So I was looking at some potential mod packs to play, and these were the ones that I found on CurseForge. I just sorted by popularity. And I looked at a review of RLCraft. It said it was really difficult because it's a super survival one. So I was looking at that one. I've heard some things about Sebtech. This one has gotten some pretty good reviews. It's like a progression mod. So I was considering that one. I don't know. Any other thoughts? What's up, NJ? Mohammed, can there be eukaryotic single-celled organisms? Eukaryotic single-celled organisms. I don't think so. Maybe. There might be, actually. Are there single-celled eukaryotes? If there are, they're not that rare. They're very rare. Oh, protozoa. Oh, yeah. Actually, you know what? They're not rare. Excuse me. I was wrong. I totally forgot about like the entire class of algae and fungus and things like phytoplankton, maybe even. Yeah, never mind. I was wrong. I was wrong right off the bat there. Yeah, there's a lot of them. I was wrong. I jumped straight to no because I'm so used to just thinking about bacteria as the only single-celled things out there. Yeah, so what was the mod pack you were talking about? You got to know your Minecraft basics. Yeah, I think so, but I was hoping I could kind of learn. I mean, I already kind of know some of the basics, but obviously not a lot because I haven't played that much Minecraft. But I was hoping I could learn the basics on the fly during a mod pack, but maybe a mod pack that's not so harsh, not so brutal on like a newer player. I don't know. Maybe I'll do some research and kind of pick one that looks the best to me. Gita, I want to answer your question there. Why do divergent boundaries create a ridge, and why are they creative in terms of oceanic lithosphere creative? Yeah, they're not creative. It's like not like they're like writing really good poetry or something. They're creative in the sense because when you think about, so let me pull up my notepad again. So like imagine you've got a like you have this tectonic plate. Let's say, let me change this color. So you've got like a tectonic plate here. Actually, it makes more sense to write this, right? So here's our plate. Actually, hold on, don't look at that. Here's our crust. And then here's our mantle. And then you've got your mantle over here too. And then you've got your other piece of crust here. Okay, so here's tectonic plate A or B. Let's say this is B and this is tectonic plate A. If they're moving away from each other, like at a divergent boundary, there's creating what's essentially like a gap in the middle, right? So what is going to fill that gap? It's the molten mantle is going to come up and fill the gap, and it's going to cool and create new crust as they're pulled apart. So what you're doing is your sort of new crust is coming up in between the plates and creating a basically brand new crust. Does that make sense, Gita? That's crazy, NJ. From all those years ago? What the heck? That was literally like 4 years ago? Okay, cool. Alrighty. Let's change this up a little bit. It's better to go on this side. So we'll just play a couple games tonight before we call it. I don't want to stay up a little bit too late tonight. Cool, banana. All right, so we almost got 10. There we go. Remember, no stream snipers. No stream snipers. Did somebody leave though? Somebody left. I think Turtlefish left the game, dropped. So I think it was just one kill. I think it was just Diaz. Okay. I don't see anything. Yeah, right. You were. Wow. Okay. Dude, I feel like green already needs to go. You can't be sus on me like that for no reason. Already green is being a little much. Oh man, oh man. Another double kill. Yeah, Arya, the game, you never played Among Us? It's pretty simple. It's basically like a game where you have a bunch of people. You've got 10 people on a ship, and you have to do these tasks to sort of fix your ship. And the entire time there's like a couple imposters, quote unquote, like people who are going around trying to sabotage the mission, and they're trying to kill people. And it's the goal of the crew to find the 2 imposters before they kill 6 of the 8 people, 6 of the 8 crewmates. Yeah, it's basically Mafia. Yeah, green is being super sus. Green is just accusing everyone. He's going hard with the accusations, which is not a good strategy if you're the imposter, in my opinion. Green voted blue. Everyone just straight got green out. I wonder if he was actually an imposter, because I got killed by blue. Could be a good strategy if blue and green were in on it together. Could be a good strategy, because green was already, it was almost like a sacrifice then if green was sort of going hard just to make that sacrifice and then vote blue so people don't think it's blue. Or green could have just been going too hard with the susses. Nope, green was going too hard. Wow. That was a good game. Yeah, good game. Less tasks. Was it too, because what did I have it set for? Short task, long task, common tasks. I'll pull it down to 2. Because I think that's what, oops, that's not the right one. I think I have it 2, 2, 2. Yeah, I think we'll try this one and see how it goes. I'm gonna play tonight until I get at least one imposter. But that could be a while because who knows how long that takes. RNG cannot be friendly sometime. First swipe. Oh wow. White quick. Electric. I did not see anything. There was a lot of people at admin though. Who wasn't at admin though? That's the thing, right? It was a ton of people. Yellow should be fine. I remember seeing yellow. Are we going black? Doing a self-report situation? Ouch, that's rough. Black. That's the worst is when like you get voted as a self-reporter when you're just like, hey, I just found a body. Everyone's like, you're sus. See ya. So blue, orange, red are in there. Electrical. Blue, orange, red. Electrical again? Who reported? Pink. So orange was there. So orange was there. Orange was in electrical, but I don't remember orange following me all the way around. So orange is a little bit sus. Could be orange. Although red is following me around a little bit, which is kind of a move that I'm not 100% about. They're saying orange is fine. Not cyan. Vote cyan. Like cyan and then pink? Why cyan though? But why cyan? Like I'm skipping because people get real quick with their votes here, and I'm not about that. If it gets down to the end and we need to make a vote for sure, but right now I think we got a little bit of time, especially if our task situation is decent. Blue is not voting. See, I abstained. I'm the only one that skipped. What's up, Pallavi? No worries, you're not interrupting. That's the point of the stream. It just kind of died down a little bit, so I just switched to play a little game here at the end before I called it a night. There was a question regarding river locations in South America. How do trade winds affect the number of rivers? That is kind of a weird question. So sometimes with the multiple choice practice tests on AP Classroom, they'll throw in, sometimes the questions are really specific and kind of odd on AP Classroom. And that's an example of one that gets weirdly specific. So what was the question? Let me pull up real quick and see what that is. because I'm not 100% sure which question you're referring to. Why? So, but it's something about wind affecting number of rivers. Looks like it's pink. Let me take a look. Question 16. Okay, I'll take a look. Phospholipid. I think, did you send me a message today? Did I respond? I think I saw you send me a Schoology message. Nice, good job. Got him. Good job, everybody else besides me who wasn't paying attention. All right, number 16. All right, everybody, give me 1 sec. 1 sec. We'll start this in 1 sec. Okay, so here is question number 16. All right, let's take a look at this one. The Atacama Desert lies to the west of the Andes in northern Chile. It is the oldest, most consistently arid place in the world. Based on the map, which of the following best explains the area of the Andes Mountains that would have the greatest number of rivers? Okay, I actually haven't looked at this question yet. I just trust AP Classroom to have decent practice tests. The Atacama Desert lies on the west of the Andes. Okay, boom, there it is, west of the Andes. Based on the map, which of the following best explains the areas of the Andes Mountain that would have the greatest number of rivers? Equal number of rivers we put on both sides because water flows downhill. Most rivers would be located on the west side because this is the leeward side. Most rivers would be located on the east side of the Andes because the trade winds are coming from the east. More rivers would be located on the south side of the Andes because it's protected from drying sunlight. That doesn't make any sense. So if I had to make an immediate guess, I would 100% guess it would be on the west side. Again, I don't know this question. I would guess it's on the west side of the Andes because of the rain shadow effect. Okay, Darsh. Thanks. I would guess it's going to be on the west side because the rain shadow effect usually creates very dry deserts on the opposite side of when the winds come in. And because this is on the coast, I would expect winds coming here carrying moisture to drop water and there to be but the desert's on that side. So then if the desert's on that side, then the rivers would be on the opposite side. Maybe it's talking about these trade winds come across the continent. So most likely probably on the east side then, if we're talking about because the wind is coming around this way and the desert is literally on the west side. Most rivers would be located on the west side of the Andes because it's on the leeward side. So whatever side the rain shadow effect is occurring on is the desert side. So that means it's this side. So the opposite side has to have more rivers, right? That's just a matter of fact. So the answer should have to be C, right? Yeah, C. Okay. Yeah. So, I mean, I think it's actually a really obvious question once you just realize, okay, if the desert's here, then the other side has to be the one that's the rain shadow effect side. Or excuse me, is the side that's not receiving the rain shadow effect. So C. Okay. Correct. Trade winds come from the east. So you have these east trade winds. I guess they kind of come across the continent of South America. My initial intuition was west because I would assume that because this is close to the coast, there'd be more wind here coming off the ocean. But I guess because of the wind directionality, it's moving this way and not coming in. So yeah. Yeah, so it's just a matter of the rain shadow effect here. If I asked you a question on the test about the rain shadow effect, it'd be more obvious than this. I would show you actually which way the wind is going right on the mountain, and you should be able to get a little bit of a better, more obvious answer to something like that. Did that make sense, Pallavi? I think once you sort of break it down, you just have to kind of recognize what the rain shadow effect is. And then that should relate to the answer there. And it was also kind of a giveaway the fact that the desert is on one side, so it would be kind of obvious that the rivers should be on the opposite side, even if you didn't know anything about the trade winds. That was quick. Orange is down. Which tectonic plate do we live on? Pacific or North American plate? We're on North American, so we actually live on the North American plate. We're on the east side of the San Andreas Fault. But remember what I was talking about if you go to Santa Cruz, you actually go onto a different tectonic plate. Okay, no worries, Pallavi. No worries. Better to be safe than sorry. Yeah, we live in kind of an interesting location from a plate tectonic perspective. I'm still just waiting for that next big earthquake. California has not been really, really rocked by an earthquake in a while. The last big one was 1994, the Riverside earthquake. We're skipping. It's kind of an interesting map here. Interesting timeline. California's earthquake timeline. Wait, Ridgecrest, 2019, 6.4? Wow. I mean, I guess there's big ones that are occurring, 6.4, 5.1, periodically, but none of these, I don't remember any of them causing major damage, like infrastructure damage that like the Northridge one in, not Riverside. When I said Riverside earlier, I meant Northridge. The Northridge quake in '94. I don't remember a major one since I've lived here, and I've lived here since 1997. White and black being a little sus. Whoa, wait, what? Who just saw that? Did anyone just see that black literally just disappeared? Really weird. Hey, Pallavi, let me see. One last question. Does the unit for review that you gave have all the topics that are going to be on the test? Yes, it does. It's pretty comprehensive. I mean, it has all the topics. I don't know if it has every single like individual possible detail, but it's pretty much, I would say, for the most part, comprehensive. Yellow or blue? Okay, hold on. Where? It's yellow and blue. Yellow on body. I was about to report. I was with purple and black. Okay, it's yellow or blue. Vote yellow, then blue. Right. Watershed stuff will not be on the exam. That's correct. It will be on Unit 5. Sorry, yellow. I'm going with what people say. Yeah, black is a little sus, but like there was no vent. You're right. So there could have been a glitch. Does that happen in the game? Like, are there glitches? Someone said they thought they saw blue vent. So we'll go yellow and then we go blue. Yeah. There could be a glitch sometimes. Vedavi. How do you pronounce your name? Is it Vidavi or Vedavi? There's green. There's yellow. Oh no, wait, I didn't see yellow. The first one. I forget which one I said first. Vedavi? Vedavi, okay. Do you have Miss Lerner? Oh, GAN. Okay, you got GAN. What is that, Adarsh? What are you saying? Peer pressured into unaliving another member of the society. Unaliving. Is that the PC word of saying... Oh no, I'm stuck with glitching black. Is that the PC way of saying killing? Unaliving. I think you can get away with saying things as long as you just add like in a video game on the end, right? Like I'm just killing people in a video game. It's not in real life. It's obviously just a fiction. Blue. Okay, so blue is clear now. This is tricky. Cams. Yeah, see, I'm not seeing anything. I'm still kind of super ignorant here about anything that's happening. Could be red. Came out of electrical and there's a vent. Green was with me. White seems to be, could be a self-report. I'm just doing it. But I don't, so the question is if it actually was yellow. Okay, so it might not have been yellow. Or it might not have been white, who knows? We just have to finish our task. We're almost done, actually. Whoa, green just super glitched through the wall there. There's red on the other side. Wait. Green. Who hasn't finished their tasks? Red, they're all here. Wait, how are people glitching around? What's happening? Is anyone seeing this? People are moving from one side of the door to the other. There's black. We're gonna win on tasks. Why emergency meeting? That's true. Black was not around while we were all around. But that doesn't necessarily mean... Wait, so I'm done. Hold on. I'm done. Black's done. Pink's done. Red is done. Wait, what? Green? Wait. I'm done with tasks. Me too. I'm done. Wait, so wait. Who hasn't finished? Oh, it could be a ghost. That's the problem though with you can't ever trust ghosts to finish their tasks. That's the issue. Oh man. So in this case, what do we got? We can kind of assume that we at least got one. Assume we got one out of here. I'm gonna skip because I don't know. Dude, red and green, yeah, we'll go against each other. Little Christmas battle here. Another question, volcanic activity is a feature of just divergent plate boundaries or convergent as well? Both. Divergent and convergent both have plate boundaries. Or excuse me, have volcanic activity at their boundaries. So we're never gonna solve this if we just start having to kick people. We're just gonna huddle up in like a little group here. So how do we resolve this if we know like a ghost that didn't finish their task left the game, right? So tasks will never get finished. So now that just means that there's no time counter. So we just have this 1, 2, 3, 4, 5. Someone took a risk. That was a big old risk there. I could not see. Holy cow. So what is it? It's 2. Although I don't know if I can I vouch for red? He's so mad. I don't know. I don't know if I can. I mean, red was definitely around me, but I don't know. Hold on. Wait, so if we get this wrong, it's not over. Brian didn't vote. So if it's not, we gotta. Oh wait, shoot! I clicked red. I meant to click black. No. I meant to click black. Shoot, that was my bad. Oh, I guess I messed up, but I still won. Oh my god! Wow, dude. That was crazy. Actually bad on my part. Actually, making a mistake actually won us the game. I accidentally voted. But I'll tell you, Diaz, you did put up a very good argument. You pled for your life there. I've never seen such pleading. Okay, we're gonna play one more game and then that's it for the night. All right, everybody, one last one and then we're out. Oh, sorry, Onslaught. Yeah, do you have to know all the types of soils? What do you mean by types? What does that mean? Later, Mohammed. Have a good night, man. What do you mean by types? Oh man, I'm not gonna get imposter tonight. Oh well. When you say types of soil, are you referring to the soil profile, like the different layers, or are you referring to like the loam? Oh no. You don't need to know all of those. You need to know, if I tell you the percent, like of the different particles, then you should definitely be able to tell me the soil type by using the triangle. So basically, if you can read the triangle with those percentages, you're good to go. But it's not like you would need to memorize all the different types, because that would just be crazy. That would be crazy. Red. Tectonic plates are found at the mantle and the crust, right? Lithospheric plates is a combo of the crust and upper mantle. Yes. NJ, this is not AP Bio. This is AP Environmental Science. Yeah. So if you look at Earth's crust. Oh, actually Earth's layers. Man, it's bad images. Can we get like a decent image here? So the crust is different than the lithosphere, and the tectonic plate is the lithosphere. So when you look at this picture here, the crust is the upper, very, very upper layer. It's what we stand on. It's where soil is, etc. But the lithosphere, which is the crust plus the upper part of the mantle, that is where the tectonic plate is. Yes. Number 10 on the practice test. Oops, that's not the number 10. This one? A model of who did they vote off? No one. A model of prevailing wind patterns around the world is shown in the diagram below. Okay, good to know. Which of the following is the best explanation for the deflection of the trade winds in both hemispheres? It's a Coriolis effect. Yeah, D, right? Yeah, the wind is deflected because of the Earth's rotation. Is there anything in particular about number 10, Geeta, that you're unclear about? Like are you unclear about the Coriolis effect and like why that causes the wind to bend different ways? NJ, I do want to play Cyberpunk if it ever comes out. I think it gets delayed constantly. I worry about the most recent delay. Because it was so close to the deadline. It was like 3 weeks away from release, and then they delayed it another 3 weeks, or like maybe a month away from 3 weeks away. But I would like to play it, yeah. I like CD Projekt Red. They make really good games typically, so yeah, we'll come out when Star Citizen does. I don't even know if Star Citizen's, that's the game that's been in like beta or alpha for like decades, right? Star Citizen, is that the MMORPG space game that was like super hyped? Man. Can't really contribute much to this because I didn't see anything. Is that okay, Gita? I hope that was clear. I think the Coriolis effect is kind of it's actually really simple, but it's confusing because it's like one of those confusing. It's just like one of those confusing things that takes your head a while to wrap around because it's not intuitive. But it's actually at its base really a simple idea or simple concept. It's kind of like, I don't know if you remember in bio when you had to learn like hypertonic and hypotonic solutions. Like really that's like a simple idea. It's just confusing because it's like there's like it's like which way is it going during with which term and like which perspective are you looking at? Like those kinds of things, like even though they're simple, they feel hard to understand because they're not intuitive. They're not straightforward per se. Sure, I can explain that a little bit. So the Coriolis effect, just sort of to verbally explain it right now, and then I can maybe pull up a diagram in a second. Ultimately, when the Earth turns, because it's a sphere, places at the equator are moving faster than places at the pole, right? Do you understand that as like a basic beginning point? Right? Like when you're on a merry-go-round, you obviously move faster when you're on the outer edge of the merry-go-round than when you're at the center. Just like when you're on Earth, a point at the pole moves faster than a point at the equator. So that's your base, right? Your baseline. So, because when you're at the whoa, why did it make it black like that? So okay, the Earth rotates this way. So I'm going to draw an arrow here. This is the way the Earth rotates. It's spinning this way. So spinning from west to east, right? So now, because a point at the equator, so let's just pick a point here. Boom. X. So this point, right, is spinning this way. Right? Everything is spinning this way. And then let's pick a point here. Okay? So if I let's say I throw something from point X to point X, right? So I want to throw something this way. Now, you would imagine if the Earth wasn't spinning, right, it would just go straight down and get to point X. But because the Earth is spinning, if where I'm standing at, let's say this is 30 degrees latitude, because I'm standing at 30 degrees latitude, I'm spinning slower, I'm moving slower than my destination is moving. So when I throw it, it's going to be carrying a slower velocity than the location it's traveling to. So what that means is that by the time the ball reaches point X will have moved further away from where the location is. So from the target's perspective, it's going to look like the ball curves like this. It's going to look like the ball curves this way because it's moving. So right, if the camera, right, is throwing a ball to me, and I'm moving faster than the camera, if the camera throws a ball right at me, I'm going to move as the ball is coming, so it's going to look like it bends this way. Right? So if I'm at the equator, air coming from north to south always bends this way. Whereas if someone threw a ball who was standing here towards me at the equator, again, I'm moving faster, so it's gonna bend away and miss me. That's the Coriolis effect. So wind is always moving from east to west between 30 degrees and 30 degrees. Right, the other 30 degrees south down here. Pallavi, yes, the practice questions are very similar. Some of the practice questions are a little bit more out there, though. Like some of the content is a little more specific and detailed, whereas the questions you'll find on the exam are going to be more straightforward, like from the actual stuff we learned, a little more like kind of like confined to that material. Okay, so Gita, I think ultimately just the way to think about it is because the equator is moving faster, right, than the 30-degree lines, the wind that's returning to the equator, because remember, due to the Hadley cells, the wind is actually coming down towards the equator from 30 degrees because of the way the Hadley cells work. And so all that wind that's coming towards the equator is bending in the opposite direction that the Earth is moving. Right? Now, in between, you got 60 degrees up here, right? In between 30 and 60 degrees, right, the wind is moving from south to north. It's moving this way because of the Ferrel cells, which are these ones right here, these spinning cells. And so as the wind is moving north, because it's coming from a place that's moving faster, right? The 60-degree spot is moving slower in relationship to the 30-degree line, because it's closer to the pole, which is the slowest of all locations. And of course, the equator is the fastest of all locations. So as the air circulates this way, its speed is going to be faster than its destination. So it's going to look like it's picking up speed in comparison to that location, right? So if I were to throw a ball towards X, because when I throw that ball, it carries the same momentum that it was traveling when it was in my hand, it's going to look like it's bending this way and actually like picking up speed. So that's kind of the best way I can explain it. Hopefully that was clear. I know it's confusing, but it's all a matter of just sort of trying to wrap your head around the idea that air is moving at different speeds in different locations, and so as it travels from north to south or south to north, it's going to bend according to how... Wait, did I just win the game by completing that task as I was totally checked out? Oh, man. Oh, man. I want to play one full game where I'm paying complete attention. But obviously my priority here is to help people with their concerns. Yeah, probably kind of like a mix of the review and the practice test. Yeah, like imagine if I took the review questions and turned them into multiple choice questions. It'd be kind of like that. It is because the Earth is globular. Globular or globular? Is there a difference between the word globular and the word globular? Or is it always globular? Okay, let's see if we can do this game here. Let's see if I can actually participate as a crewmate and not just basically just be a task performing NPC, which is essentially what I've been for the last 3 games. body. Quick body. What's up, Mitchell? I didn't see anything. I can't really participate in this one. What is ISTG? Stop sussing me. ISTG. I swear to God. Got it. Never mind. Figured it out. First, I thought ISTG was an abbreviation for Instagram. But then I realized that's not right. That doesn't make any sense. Better be, yeah, it's been full. But this is the last game anyway. Sorry. Maybe next time. I do these once a week, 7 o'clock every Wednesday. And then once in a while, I jump on randomly here and there and just do some regular old game streaming. So maybe next time. Oh, it's instigating. That would make sense. Instigating. All right. Oh, Adarsh. Oh, Adarsh, I didn't see your other question. Sorry, I skipped over it. I'll look back at it, see if I can answer it while I'm playing here. Remember how we were talking about literal truth and consciousness being a selecting force and being selected for? Would you agree that if subconscious was random, then free will has to exist? No. Why would a random process lead to free will? That doesn't make any sense. Right? Like I can roll a dice and the result is based off of randomness, but it doesn't mean I have any control over the outcome. Brown was at security. Dark green was AFK. Because a lot of people, one thing that people often bring up when you talk about like free will and like they'll talk about like, well, what about like this sort of quantum fluctuation kind of thing that generates potential randomness, and then it's just not it's not very, there's no connection there to free will. Because the thoughts aren't randomly selected for, the subconscious generates spontaneity. But your psyche, which is culturally and socially, along with biologically made up, selects for the best thoughts. Right. Yeah. No, that might be right. Like, I don't necessarily disagree with that. So you have like, so it's almost like, you're almost talking about like almost like a naturalist, like a natural select, like an evolutionary sort of process there, where you have like a random thing and then like a selecting force. You know how like in evolution you have like mutations are random, but the selection on which mutations are passed on is non-random. Is that what the analogy is you're making to like your thoughts and like the actions that you carry out based off of what you think and the choices you make? But even so, it's not. There's no decisions that you're making per se, right? So for example, like you've got like a random, like let's say you have random thoughts popping up due to whatever, like whatever you want to ascribe to it. Like let's say quantum, uncertainty or whatever. And then, oh dude, you killed me right in front of us. Dang boy, harsh. So let's say you have random thoughts popping up due to whatever, and then the selection process there is due to your brain, the way your brain works, and how it selects those thoughts based off of your culture, your genetics, etc. Again, there's no free will there for me, because it's like none of that is controlled, right? You don't control your genetics, you don't control your environment. So the thing that is selecting those random thoughts is also not part of your control. I don't think anyone else really streams, like not to the same level that I do. Mr. Gann doesn't really stream. He has a YouTube channel, but he doesn't stream. I know that back when I started doing this, Miss Lerner and Miss Chow and Mr. Hajarian would stream only when there was a test coming up as a review session, whereas I kind of embedded it as a wait, vote pink then brown. I think pink and brown. I saw yellow. Pink. Okay, I guess sometimes oops, I missed. But yeah, so I stream a little more consistently and definitely more often. Let's see. Last question before you head out, Onslaught. What happens to water at the equator? Water at the equator is heated by the sun's intense radiation at the equator. It therefore evaporates and rises in the atmosphere. And then eventually, once it reaches a certain altitude in the atmosphere, that warm, moist air cools, the water condenses and falls as precipitation back down onto regions around the equator, creating heavy amounts of rainfall in the area. Adarsh, you can control your psyche, though. You have a lot of control over your psyche, which is the controller/selector. I think you need to tell me more about what you mean, because I'm still unsure of what you're talking about. You have control over your psyche? I think my first question is, when you say psyche, what do you mean by psyche? Are you referring to, like psyche to me is a broad term. I don't know if I know exactly what you mean by psyche. Psyche is the psychoanalytical subconscious. I know what your name reminds me of. Here we go. I gotta focus here. Oh, I shouldn't have go there. Oof left the game. Okay, skip it up. Wow, that actually could work out really well. So Adar's psyche is the psychoanalytical subconscious. Okay, the psychoanalytical subconscious. So if it's subconscious, then how is it under any control? Any conscious control? Because it isn't by definition of something that's subconscious. Oh, it does legend. I didn't know that. A body's reporters, it usually tells you where it was at the bottom of the screen. That's good to know. Also, the problem is that that was my old YouTube channel. That was the one that I had back in the day. This is not the same channel. I switched channels. So I switched channels like around that time of that video, because I realized this channel that I was on was actually like a district YouTube account. So I didn't really have much control over it. So I decided to switch at that point. And I felt like it was better to have a channel that was like technically my own. Sometimes it's better to not say too much, you know? But also you want to say some things. So you don't influence it directly, but your conscious only takes thoughts that are of notable importance. Your conscious only takes thoughts that are of notable importance to it, so that the structure rests on top of the psyche. So these, and I'm not doing any work with the kills right now. Whoever this other guy's, who is just carrying me? So I'm gonna start doing some murders here. I'll be honest, I don't think anyone saw me. I can't really, there's not much I can really do to vouch. Orange is doing a good job though with how poorly I'm doing. See, I don't know what to say right now. I'm kind of messed up. I might get voted here. So Adar, sorry, I'm having a hard time, focusing on what you're saying because this game is kind of obviously taking a lot of my attention. So I apologize for not being able to have like a coherent conversation about that right now. So let me just finish this game and I'll kind of finish my last thought on that subject with you before I call it a night. I know, I keep saying that. All right, I'm going to call it a night though. So just to kind of wrap up that last combo there. Get out of here real quick. So I'm going to wrap it up here. That was some good advice. NJ, floss daily is a good advice. Let me see. I forgot what you mentioned before. Oh, tell Nyanika hi, Abhinav. Just to kind of wrap this up with you, Adarsh, I don't have time to go down a huge thing here because these conversations kind of have, it seems like they oftentimes have no bottom. I would take against your argument about bad decision making not being under the realm of criticism, since the structure we have selecting thoughts, even if it's not free will, is deterministically capable of rationalizing. Okay, so yeah, no. So Adarsh, I think you might misinterpret my. So there's a difference between like normative and prescriptive claims, right? Like, so for example, if I make the normative claim that I don't think there is free will, I'm not prescribing anything. There's no prescriptive claim there. Like a prescriptive claim would be like, therefore, you can say this for sure, or like we should do this, for example, right? See, Abhinav. Bye, Arya. So like, I would argue that in that case, it's sort of like a. I'm saying, yeah, I don't think there's not much convincing logic or evidence to support the idea of free will for me. And everything that you're saying, I would mostly agree with this idea of like potentially like random thoughts that are sort of quote unquote selected for by a environmentally or genetically or culturally shaped brain that selects thoughts based off of its sort of experiences in the world. And obviously that can change as you experience different things that shape your brain. Again, that doesn't really do anything to show free will. And so that doesn't necessarily therefore mean that we should just sort of remove all attempts at potentially like quote unquote chastising or correcting behavior that's antisocial or not productive, because of course that's not a good way to sort of carry out society. So I'm saying that we can still function without free will, because clearly we are, right? But I think there's a difference between attempting. So for example, when you say you take, a bad decision, right? Whatever that decision is, obviously that bad decision can be addressed regardless of whether we ascribe free will to that decision, right? So so someone makes a bad decision. It doesn't mean that we don't do anything, right? We still need to have norms and procedures to help address that for a variety of reasons. One, addressing that can hopefully change that person, right? Make them come to make better decisions that are better for the group or the society as a whole. And so we should look at it more as when we deal with people making bad decisions, whether they're criminals or whether they're, or it's just maybe something less severe, like just a bad decision in your day-to-day life, and you're trying to learn from that. We can understand, we can simultaneously not blame the person and still try and rectify or move to correct the decision. Does that make sense? Like the whole, like the idea of like, this person made a bad decision, therefore they deserve this sort of punishment. I think we need to look at it a little bit more. Like maybe deserve isn't the right word. Maybe it's like, it would be a good thing to correct that, but deep down we can kind of still accept the idea that they didn't ultimately have control over that decision. It was just a matter of random chance events and circumstance. And so like the idea, like a lack of free will and some sort of criminal justice system are not mutually exclusive, I guess, is what I'm trying to say. I guess. Onslaught, I didn't mention that the El Nino and La Nina originate from the South Pacific. I said that they were from the East/West Pacific. Is that still okay? Sure. Yeah. I think what you have to consider when you say east-west, just make sure you're still talking about like the equatorial Pacific along the equator near those bands. That's where the winds are originating from. Yeah, I'm thinking of doing something. So why Saturday night? A live debate Saturday night? Why Saturday night? When is so the elections are November 2nd. Oh man, we're getting close to the election. People, we're getting real close. I keep forgetting how soon it's coming up. Like, where are we at right now? It's 28th. Election ballots. The last day for ballots is November 3rd. So yeah, November 3rd. So that Tuesday is Tuesday is the election night, right? I'll definitely be streaming on Tuesday night for sure. Yeah, I think I'll stream on Tuesday night as the election's coming around. So because we're on the Pacific, we'll typically have more information at an earlier time. So I'll definitely stream on Tuesday. Yeah, we'll watch something. We'll put on some sort of we'll put some channel on, some YouTube channel that's sort of got, you know, the polls as they're coming in. I don't know what. I'll look in to see which one works the best. Yeah, so Darsh, the problem I would say is that the interpretation is selected against, since rationality gives us the ability to hijack that and say therefore the action cannot be punished since there is no fault. Right. I don't agree that you, just because there's no fault doesn't mean you can't. And I don't know if punish is the right word, right? Because punish, I think, carries with it some weird like moral condemnation. I would argue that like mostly what we should think about is like one of two things, right? Either, and this is what I think most people would argue like a criminal justice system should be, like revenge and punishment should not really be what the criminal justice system is for, right? Revenge is not really a solution to a problem. It's more of a, it's meeting out some sort of emotional response that like it's gonna satisfy you at an emotional level. Whereas I think criminal justice is probably more designed to be like rehabilitating, or if it's not possible to rehabilitate someone, let's say they have like they have a brain disorder that's just causing them to act out in aggressive and violent ways that just can't be fixed, then they need to be removed from society, obviously, and that's unfortunate. And I think that we should look at that as unfortunate rather than look at that as like ooh, that person deserved what they got. Like I think that is where I take issue with the whole like revenge, sort of like taking pleasure in revenge and punishment. To me is a very weird thing, because ultimately I think that partly has to do with the fact that I don't believe people have free will. So like how can you take pleasure in revenge or punishment when someone didn't control it? It'd be akin to like taking pleasure in punishing or getting revenge on a dog because they knocked something over in your house. Like how can you're not blamed. There's no blame to be given there. It's just an unfortunate situation. Okay. Chasing Coral. NJ, is that from the same person who did Chasing Ice? I remember watching the Chasing Ice documentary, which was really nice. Alexi, I just want to know who's going to win so I know which stocks to trade. Yeah. Alexi, I don't know. The stock market's tricky. It's weird because it could drop. I mean, the economy has taken a hit, so I would expect it to drop anyway, regardless of who wins. But yeah, I think if Biden does win, I think you can expect it to take a hit. Did it drop today? I haven't looked at it today. Oh, okay, COVID stuff. Oof, worst drop since June. Yikes. I don't even want to look now. I don't want to look at that before I go to bed. ContraPoints. I was watching a ContraPoints today. I haven't actually seen any of her videos on YouTube, and I didn't really know much about her until today. I was watching her on Dr. K's Twitch channel, talking a little bit about herself. Seems like she's an interesting person. I think I'll go watch a couple of her videos. She's from what I gathered, from what I heard her discussing with Dr. K today, was basically that she kind of tries to represent the sort of, I guess you would say, anti SJW, right? She spends a lot of time looking at like the incel community and other weird kind of niche parts of the internet culture, and like tries to like analyze them. So I'm that stuff interests me a lot, so I'm interested in that. So I'm gonna probably take a look at some of her videos. Oh, nice. At Northwestern. Cool. That's awesome. But yeah, she seemed very interesting. So I'll probably take a look at some of her videos. Can you recommend me one? You said something about justice. Okay, I'll look for one about justice. There's no control because it gives incentive to make no attempt. So Adarsh, are you taking issue with the general claim of free will? Or are you taking issue with, if we promote the claim of a lack of free will, the consequences of that, the fallout from that, where people will then sort of look at it as a way to sort of make no attempt, because they'll just sort of throw their hands up and say, well, I can't control anything, so I'll just do whatever I do. And then, and I agree. I think it might be one of those truths that might be not the best to sort of willy-nilly throw around, because it could be used in a bad way. But that's a whole different discussion, right? There's two different discussions there. There's that one, and then there's the discussion around, like, well, what about, does free will exist at all? Just from from that point of view, not the consequences. You would claim both, but just the second right now. Okay. Yeah, we definitely have a disagreement here for sure. I think we probably agree more with the second one than the first one, because the first one, I've done a lot of research and I haven't found anything that's convincing to me that free will exists, like from a logical point of view, like a logical perspective. So, but we're gonna have to put a pin in it. We'll get back on that one on another time. I want to come back on and do a Minecraft stream between now and the debate and the election. So I'll probably jump on maybe once or twice in between now. Maybe we'll see how I will see. We'll see. I'm not gonna make any promises, but one or two of the days in between now and Tuesday, I'll come on and play a little Minecraft. And I think with Minecraft, I'll be able to have those conversations with you a little bit because it's easy to pause. Yeah, go write those essays. Those are the real important things. Getting to the bottom of free will is not relevant for your life at this exact moment. All right, people, have a great rest of your night. I'm going to take off here, get some sleep. Stay safe. Stay curious. Stay all those things. All right, everybody. Peace. See ya.